Okay, so thanks, Luca, for starting the recording, and welcome to this Netpore webinar, the Netpore webinar of this month of October 2022. Today we have two outstanding speakers. First one is Ciro Chiappini. He is a professor. Sorry. Sorry, 1 second. A professor in, I'm looking for the window now. Senior lecturer in nanomaterials and biointerfaces at King's College London from 2016. His research blends nanotechnology, bioengineering, and cell biology to develop functional materials that direct cell behavior. And previously, Dr. Chiappini was Marie Curie Fellow and Newton International Fellow at Imperial College London from 2011 until 2016. He holds a doctorate from the University of Texas at Austin. In 2018, he was awarded an ERC starting grant, and Dr. Chiappini has authored more than 40 publications with over 3,500 citations across material science and bioengineering, and holds international patents. Today, he will talk us about porous silicon for advanced therapies. So, Professor Chiappini, the session is yours.
Thank you very much for this invitation, and I'm really happy to be on this seminar together with Giuseppe. I'm sure it will be a great tag team of interesting stuff about porous silicon. So, I mean, the talk is going to be about largely two aspects of porous silicon. The first one, the first half about porous silicon nanoparticles and changes to their chemistry, and the second one is going to be more about the nanoneedle platform and technology. But first, I mean, although it might not need a lot of introduction for this audience, I'd like to mention why porous silicon is a very interesting material, and it's generated by the etching of bulk silicon. Usually, you bulk etch you etch a bulk wafer. And the more conventional approach for doing that is by means of electrochemical etching. And by changing the conditions of the electrolyte, of the underlying substrate and the current, you can vary and you can tune very precisely the porous structure of the material that you obtain. And this is something that is not straightforward to do with many other porous materials that you can have. But why is it particularly interesting for use in biomedical application? And I think that the main reason is that for me is one of the few materials, or possibly the only material, that combines these four characteristics at the same time. So you have a very high manufacturability, and that comes from the fact that you can leverage all of the semiconductor industry fabrication processes and combine it with porous silicon, because it is silicon in the end. It is porous with a tunable porosity and with a very high capability of controlling it, as I've shown just before. And this allows loading and release of biomolecules of different types, going from small drugs and molecules all the way to nucleic acids and proteins. And you can, and that allows you also to control release profiles and all these kind of things, which is not simple to do. Also, because of its porosity and the manufacturability, this material can biodegrade in a controlled way. And you can very finely tune the process of degradation. And furthermore, it's very compatible. And alongside all of these things, it's a very tunable photonic material, in the sense that you can both control its light emission as well as its light absorption. And these properties are fundamental for developing biosensors. So you have to think that, well, the advantages of using this material that is potentially more expensive than, for example, polymeric solution arises is when you can combine these properties together rather than using one of them in isolation, which can be mimicked by using maybe more simpler approaches. And one of these examples, for us at least, is something that we've been starting to work on recently, which is the generation of lithiated porosilicon for tissue regeneration. And the specific tissue regeneration problem that we like to address in this instance is the regeneration of the periodontium. So the periodontium is the area that anchors the tooth to your alveolar bone, so inside your mouth. And what happens when you suffer from periodontitis is that you get bacteria that invade in this region in between the gum and the tooth, and progressively it erodes your, let me actually try to get a pointer here, and it progressively erodes your bone. It detaches your periodontal ligament to your tooth, and eventually this leads to losing the tooth completely. So there are ways to regenerate this structure, which is fairly complex and combines hard tissue with soft tissue, so the bone and the ligament itself. And it is known that for the regeneration of bone itself, silicon is a pretty good material because it induces bone formation. So you can think that using porosilicon that allows you to release silicon in a controlled fashion would be fairly good. It's also known that lithium stimulates the Wnt beta-catenin pathway. And the Wnt beta-catenin pathway is a pathway that is extremely strong regenerative signal throughout all tissues, whether they are bone or whether they are soft tissue. So it's a response to, is a pathway that is activated upon response to injury, and through the activation of axin by the inhibition of GSK3, lithium can very potently activate this pathway. In fact, this has been known for quite some time, and in the literature, a lot of people have been trying to use lithium substituted bioglasses to combine the release of silicon and lithium and stimulate regeneration in complex bone and ligament tissues. The disadvantage of using bioglass is that you can't really control the release of your ions from it. Because the process is a process of leaching. So you have a lot of release of ions in the very early stages, and then nothing else gets released anymore. And in particular for lithium, you're also limited in the amount, in the maximum amount of lithium that you can introduce in a bioglass, up to maximum 5%. But instead, the idea is that if you use porosilicon, you can actually tune and tailor the degradation of your porosilicon over time, and in that way you can control the kinetic of your ion release to what is desirable and to what is preferred in terms of regenerating your tissue. And more importantly, silicon has been used extensively as a carrier for lithium, particularly in the battery world. So, you know, porous silicon is very much used for generating lithium anodes, because it can carry up to 50% of lithium in weight, or over 4 times in molar, and you can introduce it in controllable amount. The disadvantage of this approach is that, well, actually, this material is not particularly stable when you put it into biological conditions. So if you stick lithiated silicon into any aqueous solution, it's very much likely to explode. So what we did is that we developed an approach to lithiate silicon in a way that would be biocompatible. And basically, the main difference to the process of lithiation from batteries is that when we combine the lithium with the silicon and we raise the temperature to make the lithiation occur, we control the atmosphere and we do introduce a certain amount of oxidant or a certain amount of nitride, which passivates the lithiated silicon surface. And then the material that we obtain, we go ahead and we dissolve it fully to evaluate how much lithium to silicon we can obtain. And we do that with nanowires, okay? Because they're easy to obtain, they're easy to porosify by metal-assisted chemical etching. And in that way, by controlling the lithiation parameters, we can obtain anything from 1% lithium all the way to over 40% lithium within our pore silicon. So we have a much broader range of what can be achieved from the conventional bioglasses. And we can see that we can incorporate lithium in different ways. We can either maintain the crystalline structure of the silicon all the way to the edge, and the lithium inserts within the crystalline structure, or we can create an amorphous layer on top of the original pore silicon. And regardless of whether we have only a core or we have a core shell structure, we can see that lithium is present throughout both the core and the shell of the material if we look at it by eels. Okay, and this is just the relative distribution of the silicon and of the lithium within the two core and shells structures. And indeed, by changing the parameters of the lithiation, we can control the release profile of the ions, okay? We can either have, for example, for lithium on this side, we can have a burst release where a lot of the lithium is released very early, and then we have a secondary sustained release, or we can have a more sustained release from the beginning, or we can have intermediate states. And at the same time, for the silicon, we can control the release from either being more rapid or more slow. And if you compare that to the dissolution of the original porosilicon itself, you can see that the introduction of lithium does stabilize the porosilicon matrix in some ways, because even the more quickly degrading one still dissolves more slowly than the original porous silicon. And indeed, by introducing the lithium, we do stimulate the regenerative pathways that we want. So this is evidence of stimulating beta-catenin. So axin-2 is the first gene downstream of GSK3 inhibition. And we can see that when we use this particular type of lithiation, so 1.2% lithium, which has sort of an intermediate, it does have a release, it has a 50% burst release, and then it sustains release over a couple of days. We see that if we use 1.6 milligram per ml of particles, and we put them in contact with primary human periodontal stem cells, they do activate the Wnt beta-catenin pathway at the same level as they would do if you were to stimulate them with lithium chloride. Porosilicon itself does not have any effect, nor does other formulation of lithiated silicon. Instead, if we look at pathways that stimulate bone growth or a growth of dentin, which is the other side of the mineral tissue on the tooth, we do see that both the porous silicon and the lithiated porous silicon stimulate the generation of mineralized tissue, as we would expect, because the silicon stimulus is strong in that. And in the end, if we take a mouse model where we generate a defect both in the bone and in the ligament and in the tooth, we see that with our lithiated porous silicon, we get a much better regeneration, both in terms of the total amount of bone and the density of bone that we can do with either introducing porous silicon or introducing lithium chloride, or using what is the gold standard in terms of periodontal regeneration, which is a guided tissue regeneration membrane. And obviously this improves against the non-treated control. Very importantly, we not only regenerate the bone, but if you look at this part here, we do regenerate, we form new bone, we form new cementum and new dentin here, and we do form a significant amount of new periodontal ligament at 2 weeks already. And if you look at the structure of how this is organized, you do see that the bone forms and sort of the ligament anchors into the bone onto one side and into the new cementum on the other side, which is what you would want for the regeneration of this complex interdigitated structure. Okay, so if I still have some time, I'm going to switch gear now and talk about our porosilicon nanoneedles platform. And so nanoneedles are a very interesting nanotopography to interact with cells. And the reason for that is that they have this high aspect ratio that induces a very unique and a very peculiar interaction with the cell membrane, which leads to the membrane remodeling, potentially to breaching and to higher permeability of the membrane, and to upregulating endocytosis. And because of this interaction, these materials can be used to sense what is happening inside the cells, to deliver materials from the outside of the cell to the cell, and to regulate the interaction not only with the cell membrane, but also with organelles that are beyond the cell membrane, which is not easily done with other materials, and also to record and stimulate what is happening electrically inside the cells. So we've worked on all of these three aspects. We don't really do much in terms of electrical recording and stimulation, but I will just touch base very quickly on two aspects of intracellular delivery and biosensing, and I will not really go into the biophysical interaction. So the material in particular that we use is based on porous silicon. As you can see here, is porous to 40 to 50%, and we can very precisely control through microfabrication the size of the tip, the height, and the density of these structures, so that we can regulate the interaction with the cell. Very importantly, and differently from many other type of nanoneedles that can be generated from other semiconductors or metals, because we use porous silicon, we can also tailor their degradation. And we can either have materials that are very quickly degrading, so we can use them to basically deliver very quickly, and then they just dissolve and disappear to the cell in the interaction, or things that are sort of have an intermediate degradation within a few weeks, or materials that are fundamentally long-term degrading or almost almost stable, that can be implantables and they can last for months in the body. There are essentially two ways in which you can interface the needles with the cells, and these are either by pressing them onto cells in culture or tissues, or by using them as a substrate for growing cells. And obviously this is only possible in culture. And we do both things depending on which one is most suitable for the specific application. Obviously, when we talk about tissues and things like that, you always press them onto the tissue. But when you glow cells on them, it may be more convenient to do one or the other, depending on the kinetic of what you want to do. So in terms of gene delivery and gene therapy, you have to think that when you want to use needles for delivering biomolecules, you need to very precisely evaluate what is your final goal, okay? And based on your final goal, you have to determine these 5 key aspects. So what are the type of device that you're going to use? What is the cargo, okay, based on what application you have? What is your biological system, whether they're cells in culture and or whether they are tissues in a live system, or whether they're tissues that are explanted. And that determines the way in which all of these 3 factors determines the way in which you want to interface your material to the tissue, and finally determines which are the ways in which you evaluate if your process has been successful and what assays you want to write. And we have summarized this in a recent review that is very useful if you want to get into the field and understand how practically you run experiments in this space. So one important element in the delivery is the interface. And as you can see here, and as I have anticipated, you can clearly identify that these structures do invaginate the membrane significantly, and they induce specifically extracellular vesicles and endocytic vesicle formation around individual needles. And we think that these play a significant role in the delivery of cargo from the needles to the cells. And indeed, when you interface cells in this way and you get these significant membrane deformations, you end up having significant delivery of nucleic acids into the cells. And in this case, we were doing it with a plasmid that was coding for the GFP protein and an sRNA that was coding that was targeting GAPDH and was labeled with Cy5, and we were co-delivering them from the needles into cells. And we could see that you can get up to 90% efficiency in the delivery of both the sRNA and the GFP. But you're not really limited to what you can do in vitro. I think that what is more interesting is the ability to nano-inject topically to specific locations in vivo and deliver genes that way. And you can clearly see that when you use nanoneedles, you can very well define the region in which you want to deliver, differently from whether you just drop your payload onto the animal or whether you are using a flat chip that does not have needles to deliver. And more importantly, if you deliver with these 2 approaches, your payload gets washed away immediately at time 0. But if you deliver with nanoneedles, you can retain the payload at the site of delivery for up to 48 hours and actually even longer. Obviously, this is something that you can do with microneedles as well. But the microneedles have a very precise way of delivering only in the vicinity of the individual needle itself. And there is 1 needle every hundreds of microns. And that means that in practice, you are delivering a lot of your payload in very few areas within your tissue. And that's not very efficient if you want to transfect something to the cells that are existing in between these very concentrated delivery sites. So you end up having a very high gradient of your delivered payload. Instead, when you deliver with nanoneedles, you see that you have a much more uniform distribution of your payload throughout the area of the tissue that is interfaced. And indeed, when we did these to the muscle of an animal, and we delivered the plasmid that was coding for the vascular endothelial growth factor, which stimulates the generation of new vasculature, we could see that over the course of 2 weeks, the animal did grow new vasculature that was attached to the original vasculature of the animal. And this new vasculature growth was driven by the expression of the gene that we had inserted. And this was just a proof of principle that we could actually do that. But since then, we've moved into using this approach of delivering nucleic acid topically to tissue to address specific diseases. And one of them is corneal endothelial dysfunction. And what happens in corneal endothelial dysfunction is that you lose the cells that die off in the inside of your cornea. And these cells don't regenerate because they don't have a capability, they are senescent, and they can't reproduce themselves anymore. And when you lose them, you can't control the ingress of water and fluids within your cornea. The cornea swells and you lose this clarity and you lose vision. Okay. So the idea is that if you can regenerate these cells, if you can stimulate these cells that are still there to reproduce and to divide, then you can reestablish the endothelium and control water ingress. And so what we did is that we loaded our needles with a siRNA that targets P16, and P16 is an inhibitor of cell division, okay? And then we've taken the needles and we have applied them directly to the endothelium of a human cornea. And you can see here that the nanoneedles interface directly with the cytosol of all the cells within the endothelium. And they can deliver the siRNA that is labeled in red within this corneal endothelium. And when we deliver the siRNA from the needles, we can actually downregulate the expression of P16, okay, which then leads to these cells going back into the cell cycle and reproducing. We can clearly see that when a cell has been delivered to with siRNA, it stops expressing the P16, which is labeled in green. Okay, and then we can think these are as an intervention to apply directly to the inner cornea. We also apply these to skin wounds in epidermolysis bullosa, which is a genetic disease where people lose the expression of collagen 7, and this generates a delamination of the epidermis from the dermis, which first generates this blister and then these open wounds, which end up becoming chronic and then becoming squamous cell carcinoma. Because this is due to the loss of expression of one single gene, what we can do is that we can load these individual gene onto the nanoneedles, and we can transfect it directly to the fibroblast, to the skin fibroblasts that were originally not expressing these protein, so that they express it. And as you can see, we get over 60% of the fibroblasts to express the genes, whether we do it with either a rigid chip or something that we are developing now, which are bandages that contain our nanoneedles. So I will very briefly, well, okay, if I am I over time? Then I can probably just briefly introduce this system where we combine nanoneedles with a CRISPR-Cas12 sensing or reporting system to sense ATP intracellular. And I will just very briefly explain how that works. So the nanoneedles gives us intracellular access, and we decorate the needles with an locked aptamer that locks an activator for the CRISPR-Cas12 system. When the aptamer recognizes ATP, it gets released and it exposes the activator. We then extract the needles from the cells, and so the cell can continue to be alive in culture, and we expose it to the Cas12. And the Cas12 then indiscriminately cleaves a fluorescence quencher pair, and then it generates fluorescence based on the original amount of ATP that we had in the cell. And we've done these in live cells, and we can see that when we apply the needles from the top, and then we use the system to detect the cells, we can detect the amount of cells that we have, okay, whether we have 30,000 or 50,000 cells. And we can also quantify the ATP concentration, which is around 1 millimolar, and the cells that are there remain alive, okay? And then we'll just forget about that. So I hope that in conclusion, I have proved that porous silicon is a versatile material for delivery and sensing, that we can use porous silicon to stimulate the regeneration of the periodontal ligament, and that nanoneedles can be used for gene therapy in vivo, and that we can also use them to monitor the intracellular conditions with minimal invasiveness. With this, I'd like to thank my group and our collaborators, our funding agencies. And if you're interested in nanoneedles or in general nanotechnologies for cell manipulation, I invite you to join us at this symposium at the MRS in the MRS spring. Thank you very much for your attention.
Thank you very much to you, Professor Sciappini. It has been really impressive. It's a pity we only have an hour for these short webinars, but now very short time if there is any question.
I have a very short question, Giuseppe, for Ciro.
Go ahead, Giuseppe.
Yeah, Ciro, very, very nice work. Thank you very much. Yeah, I was just wondering if you have a number about, if any, the percentage of cell you kill when you do the intracellular sensing, or there are no that cell.
I think that for the intracellular sensing, you don't kill a lot of cells. Okay, what can happen more often is that you pick up a few cells. Okay, and that is a bit more difficult to quantify, and it depends a lot on what type of cell you're using and what type of sensor you have developed. So different sensors have different affinity to pick up cells.
Good, thank you.
Thank you, Giuseppe.
Giuseppe,
Yes.
I have a question.
Go ahead, Luis.
Yeah, Ciro, thank you for your presentation. Very nice results and very impressive investigation. So I have a question about the lithiated porous silicon, about the degradation of the porous material. So have you trying to analyze the effect of degradation under different pH conditions? So you are using for promote the cellular response. So do you have any idea about what is the effect of the degradation of lithium in porous silicon under different pH conditions?
Okay, so to answer to that, we have not checked the degradation at different pH because it was too much to just do it at pH 7. What we do know is that the degradation does not affect the pH of cell culture medium. So in other buffers, it might affect it, but when we put it in cell culture medium, we make sure that the pH remains balanced because that could be harmful for the cells.
Okay, thank you very much.
Okay, we thank again Professor Ciappini, and we hope we can follow these works now better with all these ideas. And now it's time for the second speaker of this webinar, which is Giuseppe Barilaro from University of Pisa. He is currently associate professor at the Information Engineering Department, and he has been leading in this department his own research group, where the primary research interest is the preparation of micro and nanostructured materials, devices, and systems with applications to nanophotonics, microelectronics, biosensing, and nanomedicine. Professor Barilaro is author of more than 300 scientific publications, among which 104 papers on peer-reviewed international journals and 20 patents, 10 of which granted in USA, Europe, and even China. So let's welcome now Professor Barilaro. Thank you, and it's your time. Please, Giuseppe, you should activate your microphone.
Okay, can you hear me now?
Yes.
Okay, great. Can you see the chart?
Yes.
Okay, great. Okay, yeah, thank you very much, Giuseppe. Thank you everyone for being here, and the Netpore for this kind of invitation. I'm really happy to talk about some of the research stuff we are doing on porous silicon. And yeah, today I'm going to talk about, I try to, yeah, sorry about that. Okay. About the use of porous silicon for imaging and biosensing. So this is a short outline of my talk today. I'm going to briefly introduce the University of Pisa and my group, and then I will discuss two specific applications we are working on in my group. The use of porous silicon for the fabrication of photonic and plasmonic microlens for enabling fluorescence microscopy with the smartphone. And also a brand new activity we've been carried out over the last few months on the development of a bioresorbable chemical sensor for in vivo applications. So as many of you know, the University of Pisa is here, where is the Leaning, where you see the Leaning Tower. So we are halfway between Rome and Milan. It's a pretty small town, but a pretty large university. We have 60,000 students, and the town population, it's about 100,000. 120. So half of the population is basically students, and yeah, we have a very nice countryside, and we have the river, a very enjoyable sitting, and you can just walk from one side to the other side of the town. Yeah. This is old picture of my group. Is not actually updated, but what we do basically is playing with the nano and micro materials. So we do quite a lot of different stuff using polymer or metal printing. For example, we have a nano needle printed out of metal here. And we do microneedle with joint technology with organic and inorganic materials. So what I'm going to talk today, it's however about porous silicon. And as Ciro explained very well in his talk, porous silicon is an amazing materials because you can just fabricate very easily by electrochemical etching of a crystalline silicon substrate. You can tune the the etching parameter to control the porosity. At the nanoscale, we cannot really control the pore position, but we can control the porosity. So the mass of material removed per unit time, and this value depends basically on the etching current density I'm flowing through the electrochemical cell. So by changing the etching current density, I can change the mass of materials removed per unit of depth. And then if the current density is constant over time, I get straight pores. But on the other hand, if I switch the current density between different values during the etching, what I get are two materials with different morphology and then with different porosity. And this means that these materials have different reflective index also, which means I can control the light propagation within these materials in a very easy way. For example, by tuning the periodicity of the etching current density and then on the resulting structure, you can see we can nicely tune the color reflected by the sample. These colors are called structural colors, and have been using a lot in nature. For example, there are beetles. If you look, for example, at the shield of this beetle, it looks like exactly the porous silicon sample I show you now in this chart, and the color comes from constructive and destructive interference effect or white light with this periodic multi-layer structure. So in my group, we try to think about how can we leverage porous silicon to push some of the functionalities of a smartphone, for example. So I'm sure you know that the smartphone we use daily in our life as a computational power, which is 1 million times larger than the computer used to drive the Apollo mission. And so there is a lot of power in our smartphone, and so we thought to leverage the porous silicon to try to fabricate simple lens that could help to transform a smartphone into a fluorescence microscope. And for this reason, porous silicon is very smart material, because you can really tune the morphology of these materials over depth. For example, in this way, in this video here, you see one of my students dropping a controlled volume of silicon, liquid silicon, on top of a porous silicon layer, and the silicon, instead of spreading over the surface, just form like a hemisphere, an hemispherical object, like a lens. And in this case, we just leverage the possibility of controlling the morphology of the porous silicon during the fabrication to do a nanostructuration on the surface like a lotoflower. The lotoflower works for water. In this case, our nanostructuration works for silicon, but the concept is exactly the same. And then if you look, let me see, laser pointer. If you look here, and the magnification in this region between the top layer and the bottom layer, there is a stop layer with very low porosity, because in this way we avoid the infiltration of the silicon underneath. And then underneath the stop layer, we can fabricate an optical structure. In this case, it is a filter, an optical filter. So the nice thing here is that after you form this lens, you can etch away the porous silicon around the drop, because everything is passivated, is protected by the silicon. And then when you peel the lens out of the silicon chip, you get the lens, and you get the lens as the optical filter integrated in it. So in this way, you can easily fabricate a magnification lens which has an optical filter integrated in it. And the nice thing with the silicon is that silicon can be used to, it's very adhesive to glass surface. So you can stick several times, as many times as you want the lens to your smartphone camera and use it for imaging. For example, in this picture here, I show you the magnification of this tiny structure here in the red box using a lens attached to a commercial smartphone. And you can see here that we are able to nicely resolve a few micrometer with the smartphone. So basically, we are transforming a commercial smartphone into a microscope. But don't forget, we have a filter integrated in the lens. With the porous silicon, we can just tune the optical properties of the filter. For example, we can integrate a filter working in the blue region or in the red region or in the green region. In this way, we change the light passing through the lens, and then we change color and we can filter out some specific color. For example, here you see a few lenses with different optical filter integrated in the lens. And this is the image, a picture of Marie Curie. The image is very tiny and it's in black and white. But then if you use a lens with the pure silicon filter to image this tiny picture, you can magnify the picture, and then you can change color to the picture because you filter out some specific color from the white light reflected. So what is the main application? One of the possible application of this lens. So the lens is very low cost. So it costs like 1 euro cent to fabricate. And if you integrate a correct filter like this one, for example, to filter off the blue light or the UV light, you can use to transform the smartphone into a fluorescence microscopy. For example, here you see an image taken with the smartphone equipped with a lens containing a porous silicon blue filter. You see basically the lens operate where you see the black spot, and the green light is the laser light leaking through the lens, to the edge of the lens. But in the central area, you clearly see the tumor cell, and you see the fluorescence, the red fluorescence of the tumor cell, which has been excited with the laser. So basically, the filter can cut off the excitation line, and so the lens can recover, can magnify the image and can recover the fluorescence of the cell. In this way, by integrating a different filter on different wavelength, for example, you can always cut off the laser light, but then recover different colors, also different fluorescence colors. In this way, we can distinguish, for example, between dead tumor cell and live tumor cells. And in this column here, you see the image acquired with the smartphone equipped with the lens. In the right column, you see the same image, but acquired with a commercial fluorescence microscope. Of course, the quality of the image are different, but you can still do a cell counting. And if you think that the difference are about 20,000 euros, so I think that's a good result. Yeah, we can do more. We can do the same process, but then we can coat the lens with by just putting the lens into a solution of metal salt like silver fluoride, and then so the lens is coated with the silver nanoparticle. You can do the same with the gold nanoparticle or whatever. And this means that you integrate on the lens surface also a plasmonic filter that cut off again the light at some specific wavelength depending on the resonant, on the plasmonic resonant or your nanoparticles. In this way, you can, let me remove the pointer. Okay. So in this way, you can, for example, use the smartphone to image protozoan in just a drop micrometer inside. So the shield is silica, and some of these protozoa they are just living and swimming in the water, and some of these protozoa eat like a microscopic algae, which are somehow fluorescent in the red. And then again, you can image in the bright field on also in a fluorescence microscopy the protozoa swimming in the water. And this is very interesting because the protozoa, this is the good cousin of the protozoa and the malaria, for example. So this means that you can really do live microscopy. And of course, you can also do a counting of the cells. Okay, I hope I convinced you that you can really easily fabricate magnifying lens with the photonic or plasmonic filter embedded in your lens. And the quality of the lens is not affected by the filter you integrate in the lens, because the filter are nanoscopic and the lens is somehow macroscopic because as a millimeter size. Yeah, I would suggest you to try to do the experiment. It's really easy to fabricate this lens for your smartphone, and you can just enjoy using it. The last part of the talk, in this last part, I would like to talk about a new activity we've been carried out over the last months. We've been funded with a €3 million project by the European Innovation Council, and the project have just started a few months ago. And the objective of the project is to go from ex vivo sensing to in vivo sensing, meaning that we want to change the paradigm here and not bring the sample from the patients to the sensor, but bring the sensor into the patient and do monitoring of analyte of clinical interest in vivo. Yeah, basically the final goal of the project is to develop a chemical sensor system which is implantable, biodegradable, and allow continuous monitoring of chemotherapeutics drugs in vivo. So we just have some preliminary result, and we started from our first know-how on biosensing. Basically, we've been using biosensor, porous silicon, to fabricate biosensor for ex vivo application. In this case, for example, we took a porous silicon layer, and then instead of relying on the typical chemistry for silicon functionalization, we came out with this new approach, which is the layer by layer functionalization with the polyelectrolyte. The advantage of this approach is that you can use the polyelectrolyte perform a conformal coating of a very high aspect ratio structure. So we are able, with the single layer of polyelectrolyte, to completely coat all the pores of the porous silicon layer up to several micron depth. But the nice thing is that also we can do the synthesis of polyelectrolyte to carry already a bioreceptor. In this way, we don't have to put the bioreceptor after the functionalization, but we engineer the polyelectrolyte with specific bioreceptor, and then we do the assembly in the porous silicon. In this way, we know the amount of bioreceptor we bring into the porous silicon sensor. And in this case, for example, in this first paper we published a few years ago, we demonstrated that it was possible to really push the detection limit of porous silicon biosensor with this functionalization approach. We got a limit of detection of 100 femtomolar roughly for the streptavidin. Yeah, sorry about that. With very good selectivity against pepsin and BSA. So we thought it is possible. It is a good start to carry on with this approach, because this approach it works well, and also as we have to go in vivo, the porous silicon is a natural filter that filter out most of the unwanted molecules that you can find in the body, like cells, fat molecules, or bigger molecules that or molecules that are bigger than your target analyte. In this way, with the sensor I showed you before, for example, we were able to do measurement of streptavidin in saliva, in raw saliva, with very, very nice result. So we started with the porous silicon, and then we do the oxidation, the porous silicon, and then we transfer print the membrane, the porous silicon membrane on a PLGA substrate. This is because we want to go in vivo, and then we want to be bioresorbable, so we cannot implant the full silicon chip. So the membrane, the porous silicon membrane, is just 5 micrometer thick, and the PLGA layer is also 10 or 20 micrometer thick. Again, in this case, when we go in vivo, we don't want to rely on a label-free approach. It would be difficult to monitor refractive index change in vivo. So in this first proof of concept, we demonstrate fluorescence monitoring of pH in vivo. And to do this, we just modify, we use the same approach, biofunctionalization approach. We use a different polyelectrolyte, and we modify the polyelectrolyte with fluorophores. So basically, the polyelectrolyte we assemble between inside the porous silicon scaffold are fluorescents. But the fluorescence of the polyelectrolyte depends on how close the two polyelectrolytes are to each other, because if the polyelectrolytes are very close, there is a self-quenching of the fluorophores. And if the polyelectrolyte swells, and so the distance between the fluorophore is bigger, then the self-quenching decreases. So the intensity is basically modulated, the fluorescence intensity is basically modulated by the pH, because the pH changes the ionization degree of the polyelectrolyte, and then change in turn the electrostatic force that brings the polyelectrolyte together. So depending on the pH, this force increase or decreases, so the polyelectrolyte shrink as well, depending on the pH value. Here you see some of the fabrication result, the membrane we detach, and in this picture here, you see the PLGI foil with the porous silicon membrane functionalized with the fluorescence polyelectrolyte. And the nice thing of using porous silicon is that we can really leverage the increase of the specific surface of these materials. In fact, we can increase the the photoluminescence intensity of about 500 times compared to the flat substrate, which is important for in vivo application, because we need to go through skin. And so what we want to do is measure the change in the fluorescence to skin. And this is very, very, very useful for that. And then here you see some of the in vitro results we get on pH sensing. You see the fluorescence is modulated with the pH level, and then you see we have a very linear, nice linear calibration curve over pH. I want to highlight that usually fluorophore, if you do pH sensor with single fluorophore, the single fluorophore allow you to monitor 1.5 point of pH. But in this case, we can easily go in a linear way from pH 4 to pH 7.5. This is because the fluorophore is not sensitive to the pH value itself, but it is insensitive to the pH value. It's the multi-layer, the polyelectrode multi-layer, which is sensitive to the pH. You can see here that the reliability over time, so this is a time resolved PL curve versus pH value. You can see when the pH decrease, the intensity increase, and then when the pH goes back, the intensity goes back, and there is no significant hysteresis. We demonstrate that it's possible to monitor the sensor, not just taking a spectrometer, but also using an LED and a photodiode to measure the integrated intensity. And then we can use a simple electrical circuit to convert the photodiode intensity into a voltage over pH with good results. But then the main focus is that we have to go in vivo and to be biodegradable. And here you see, for example, this is an experiment we carried out with synthetic skin. You see the PLG foil and the porous silicone membrane with the polyelectrolyte. And then we carry out the experiment over 250 hours. You can see basically that the fluorescence, when the pH is stable, is still stable for 110 roughly hours. And if you, at a given point, like after 50 hours, you change the pH from 4 to 7.5, you can get a very nice calibration. The calibration curves doesn't change significantly over time until 100 hours. And then after 100 hours, roughly, the silicon scaffold starts to degrade and disappear in an hour, roughly 100 hours. And here you can see a nice demonstration about this. Here you see after the experiment, after 4 days, you lift up the upper layer of the skin, and you see that you just find the PLJ, and the sensor, the porous silicon sensor, almost disappeared. So this is nice, but then we have to go in vivo. We went in vivo. We did the several experiments with an animal model. We implanted the mice with the sensor on the skin, and then we measured the real-time change of pH induced on purpose. You see, we can clearly see the increase of the fluorescence when we inject, we change the local pH from physiological pH to pH 4, and then we can measure also the change in the intensity in real time. And in this figure here, basically, you see the result of three different mouses. We double check also the biodegradation in vivo. For example, you see the sensor is still there in the first day, but after 1 week, we don't see the sensor anymore. And then after 2 months, we haven't found any residual of the sensor. And then after 2 months, we did organ explantation. We double check any possible biocompatibility issues in the different organs, from brain to kidney, and also in the skin, and we haven't found any trouble with that. So basically, this was our first work on bioresorbable chemical sensor. I think it was actually the first work on bioresorbable chemical sensor using porous silicon. And the results are very encouraging. We are now working on the sensing of chemotherapeutics, and yeah, that's it. I think this is a nice trend, and would become very popular in the future if we manage to demonstrate that it's possible to fabricate sensor for in vivo applications. Let me thank the collaborators of the work I show you, Professor Max Sedor, Dr. Dan from Software Nanotech, Professor Giuliani for in vivo experiment on the biodegradable sensor, and Professor Di Giuseppe for providing the protozoan for imaging, the RESOL project for the funding, and also the other sponsor, and all of you for your attention. Thank you very much, and I'm here if you have any question.
Thanks, thanks Giuseppe for this very nice presentation and for the excellent timing. So we have time for a few short questions, if any.
Yeah. Hi, Jeff. Really interesting. I really like the biodegradable sensor, particularly the lens. I had seen it before, so more curious about this last one. Because it relies on the swelling of the polyelectrolytes. Do you have any limitation on the number of cycles that you can undergo with these, or on sort of the strength of your porosilicon matrix with it, or it just works perfectly?
No, what we have seen, Ciro, is that the sensor is really reliable. I mean, you can cycle the sensor as much as you want until unless we cycle the sensor for a few days, for 4 days continuously, we haven't seen any aging effect. But the limitation here is the degradation of the porosilicon scaffold, basically. So I think I have some backup result depending on the oxidation. We do the oxidation, and depending on the oxidation degree, you see that the scaffold degrade in a different over different time. So once the scaffold has been degraded, well, the nice thing is that once the scaffold has been degraded, you see here the degradation of the scaffold, the multi-layer, the polyelectrolyte multi-layer still works for at least 50 more hours and then degrade. So this is interesting because basically it doesn't mean that the porosilicon is just a scaffold to increase the amount of polyelectrolyte you can bring, but then the polyelectrolyte works by itself, the multi-layer.
Thanks. Very interesting.
Any more questions?
Yeah, yes, I have a question, Giuseppe. So,
Luis?
Giuseppe, thank you for your presentation, nice presentation and great results. So I have a curiosity when you comment about the functionalization of porosilicon with the polyelectrolytes, you get a very, low limit of detection for biotin and streptavidin. So this is the structure is a monolayer of porosilicon, or you are using a multi-layer with photonic structure?
No, this is just a monolayer. The advantage of using this polyelectrolyte is that you can really control the coverage in a much better way. For example, this polyelectrolyte have not been used before for sensing, but they have been largely used for drug delivery for silicon nanoparticle. So you can really increase the amount of bioreceptor you can get inside the porous layer.
Okay, thank you, Giuseppe.
Okay. There are any more questions? If not, I think we can thank again both Professors Chiappini and Barilaro, and also thank you for your attendance to this webinar. And okay, we see us back in the following webinar. We hope again with Professor Kunin. Thank you.
Thank you very much. Have a nice day.
Thank you. Bye-bye.
Bye.
See you.
Bye.